This is the second lecture of the embryology, and title is Cell Division and Cell Cycle. So there are two types of cells, the sex cells or germ cells, which are present in the gonads, ovary, or the testes. Second is the somatic cells, which are present in other parts of the body. So both type of cells, they divide. There are every cell have its cycle, and there are different the periods of the cell cycle. So either the cell is dividing phase, or in the resting phase, not dividing. The resting phase are also known as the interphase. So the resting phase doesn't mean that nothing is going to be happen during this phase. It is further divided into three phases. The first is gap 1 period. The second is synthetic period as S phase, and gap 2. So during gap 1, this RNA synthesis and protein synthesis occurs, and during synthetic phase, DNA replication, and there is the doubling of the chromatin material and chromosomes. And then during gap 2 period, it is now ready to divide, and it becomes parent cell, and at this place the somatic cells, they have 46 double structured chromosomes and 4N DNA. Double structured chromosomes mean there is one centromere, and the 4 limbs, and this is known as double structured, and this is centromere, and 2 limbs, this is known as single structured chromosome. So at this place, just before division, the cell is known as parent cell, and the 46 double structured chromosomes and 4N DNA, that is the double DNA. On the basis of the cell cycle, there are 3 types of cells. One is the permanent cells, means that once it is divided, then it comes out from the cell cycle, and now there is no further division. Such cells are known as permanent cells, and these are the nerve cells and cardiac cell, and some say that the skeletal muscle cells. Second type, stable cells. In normal condition, they don't divide, but once there is stimulus given to the cells, they start dividing. Like there is injury on the skin, so the cells, they start dividing to bridge the gap. In the same way, If there is injury to the liver, so these cells start dividing, and when it is healed, so there is no cell division. And third type is the labile cells. They continuously divide on a slow pace as the intestinal cells, also the skin cells. They are two types. They are dividing on low pace, sloughing on the surface, and then growing from the basal layer, and it is also known as stable cells because if there is injury, they start dividing on the greater pace and speed. That is known as the stable cells and labile cells. They are continuously dividing and dividing. So, on the basis of the cell cycle, there are three types of cells: permanent, stable, and labile cells. For the cell division, there are two types of cell division. The one is the mitosis. It is also known as typical cell division, and it occurs in almost all type of cells, and the most the commonly occurring. So this occurs in all the somatic cells. And the early phases of sex cells, then all embryonic cells. So now see all embryonic cells in after fertilization. So division is through mitosis, and also in the sex cells, all somatic cells. So the most commonly occurring cell division is mitosis. So there are four phases of the mitosis. Number one is the prophase. Number two, metaphase. Number three, the anaphase. Number four, telophase. During prophase, four events occur. Number one is the visibility of the chromosomes. Chromosomes, they are visible. So during the cell resting phase, they are not visible under light microscope but can be seen under electron microscope. So during prophase, the chromosomes, they are visible. Second is esters and mitotic spindle formation. Mitotic spindle formation. These are formed from the esters are formed from the centrioles, and mitotic spindle through the macrotubules. Then nucleoli disappear. Nucleoli disappear. And last is the rupture or degeneration of the nuclear membrane. This is the end of the prophase. Then next is the metaphase. After rupture of the cell nuclear membrane, the double structured chromosomes, they are floating in the cytoplasm, and then during the metaphase, these chromosomes, they are attached on the widest part of the mitotic spindle known as equator, and you know that there are 26 double structured chromosomes. So there are 46 the points where these chromosomes are attached to the mitotic spindle. During anaphase, the now note that there is splitting of the centromeres. During anaphase, there is splitting or breakage of the centromeres. Now what happens? So from one chromosome, two single structured chromatids are formed after breaking of the centromeres. So in anaphase, what happens? There is splitting or breakage of the centromeres, and the two chromatids or single structured chromosomes, they are formed by splitting of the centromeres. So 46 single structured chromosomes, they move towards one pole, and 46 they move to the second pole and gathered at the ends. So during telophase, that the reverse of the prophase, so what happens? Formation of the nuclear membrane, then the appearance of the nucleoli, then disappearance of the mitotic spindle and ester formation, then the chromosomes, they become invisible, and in the result two daughter cells are formed, each having 46 single structured chromosomes and 2n DNA. And these chromosome and genetic material is same as the parent cell. So these the daughter cells are identical to the parent cell. So though no genetic change, and in this way now these cells, they enter into the interphase, and again there is the three phases: gap 1, synthetic, and gap 2, and they are ready to further division. It is known as atypical division, and it is the less commonly occurring. It occurs only in the late phases of sex cells. So it is divided into further two divisions. The first meiotic division and second meiotic division. The parent cell, the same parent cell having 46 double structured chromosomes and 4n DNA. So this cell now enters into the first meiotic division. And now what happens in the first meiotic division? Again there is prophase 1. So this prophase 1 is the longest phase of the first meiotic division, or even all the divisions. So it is further subdivided into 5 phases. The leptotene, zygotene, pectine, diplotene, and diakinesis. During leptotene, the visibility of the chromosomes, and zygotene, the pairing of the chromosomes. 23 pairs are formed. 23 pairs. So 2 chromosomes, they lie close together to form one pair. In this way, 23 pairs are formed. 22 are autologous, and one is the this sex pair, heterologous. So now pairing occurs during zygotene phase, then during the pectine, crossing over of the chromosomes. Some part of the chromosomes of the same pair, like this, is another chromosome. So these parts, they are crossed over among the two chromosomes of the same pair. Now these cross parts, they are detached from the original chromosomes, and they are attached to the other chromosome of the same pair, like this. So here they remain united. So these parts, these segments, they are exchanged between the chromosomes of the same pair. In this way, there is reshuffling of the genetic material occurs during pectine. During the diplotene phase, these chromosomes, they start the separating from each other, and X form shaped or the plus shaped parts, the figures, they are formed, and these parts are known as the chiasma formation. chiasma formation occurs during the diplotein phase. Another three changes of the prophase, like the formation of the mitotic spindle and the ester formation and the this disappearance of the nucleoli and rupture or degeneration of the nuclear membrane occurs during diakinesis. And now in the leptotein, visibility of the chromosomes, zygotein, pairing, pachytene, crossing over and exchange of the genetic material segments, diplotein, the chiasma formation, diakinesis, the other three. So this phase, diplotein phase, is the longest phase. It can extend up to 40 or 45 years. Longest phase. Now next is the metaphase. So what they note the difference between the metaphase of the mitosis and metaphase of the first mutic division. Here the pairs, they are arranged or attached to the equator of the mitotic spindle, not single chromosome. Pairs. So there are 23 points. In the mitosis there are 46 points. Here 23 points where these pairs, they are attached to the mitotic spindle. 23 pairs, each having 2 chromosomes and 2 centromeres, but all are double structured. What happens in the anaphase? So again, note what the difference between the anaphase of the mitosis and anaphase of the first mutic division. There is no splitting of the centromere. Mind it, there is no splitting or breakage of the centromeres. What happens? The 2 chromosomes of the one pair, one chromosome move towards one pole, one chromosome move to the other pole. So 23 double structured chromosomes, they are gathered at one pole. 23 double structured chromosomes, they are gathered on the other pole. So there is no splitting of the centromeres. So now next is the telophase, and the 4 things happens. The nuclear membrane is formed, then the nucleoli appear, and the mitotic spindle disappears, and the chromosomes become invisible. Now at the end of the first mutic division, 2 daughter cells are formed, 23 double structured chromosomes and 2n, 23 double structured chromosomes, 2n. Now second mutic division starts without interphase. Now see, there is no interphase between first mutic division and second mutic division. So no interphase. These, they directly enter into second meiotic division. So now these are the parent cells for the second meiotic division. Second meiotic division, it is just like mitosis, except there are 23 chromosomes taking part. In mitosis, 46 chromosomes, here the 23 chromosomes, but the events are the same. So first is the Prof phase 2. Four things: visibility of the chromosomes, then mitotic spindle formation, then the disappearance of nucleoli, and rupture of the nuclear membrane. Now in the metaphase 2, so now see now single chromosomes, but they are double structured. They are attached on the equator of this mitotic spindle, and now see there are 23 points where these chromosomes are attached, and each the single chromosome double structure. They are attached on the equator of the mitotic spindle. Now anaphase 2, now same as mitosis, splitting of the centromeres occur. Splitting and breakage of the centromeres occur, and the single structured chromosomes or chromatids are formed. So the 23 single structured chromosomes, they move towards one pole, and 23 single structured chromosomes, they are gathered, are moving toward the other pole. So this occurs in the anaphase, and then telophase 2. So the four things reversed: nuclear membrane is formed, nucleoli appear, mitotic spindle disappear, and chromosomes become invisible. Now four daughter cells are formed, each having 23 single structured chromosomes and one N. Is a haploid number of chromosomes and haploid DNA. So among these, in males, these two have 22 plus Y, and the two have 22 plus X. In female, all are of this type, 22 plus X. So at the end of complete meiosis, first and second meiosis, four daughter cells are formed from one parent cell entering into the first mutated VN. So the four daughter cells are formed, and the C chromosomes are not identical to the parent cells, as there is some reshuffling of the chromatin material.